Hi students, today we are going to begin with a new chapter, that is biotechnology, the principles and processes. Very interesting topic. And the topics to be covered in this chapter's introduction, principles of biotechnology, tools of recombinant DNA technology and processes of recombinant DNA technology. So what do you mean by biotechnology? We discussed earlier about plants, animals, gene, genetics, etc. Right? So what do you mean by bio? Bio means something which is related to living things, life. Then technology, tech. Yes. So what do you mean by biotechnology? So simply it's a combination of bio and technology. That means life or living things and technology. Or simply we can define it as advanced technology which is used in relation to living organisms. Which is useful or non-useful? Definitely which is useful for human beings. We human beings, whatever we develop, we mainly focus on what is benefit in them, right? So definitely this biotechnology is benefiting a lot to the human beings. And it is a field which is an upcoming field which is going on, research work is going on done in this area of biotechnology. So lots of research work is being going on in the biotechnology and it's a very upcoming field also. And today we are going to discuss in detail about the biotechnology, the introduction. And what is biotechnology? Already discussed, that is biotechnology is the technique of using live organisms or their enzymes for products and process useful to human beings. So we know already what are the different technology which actually we are traditionally already we've been used in our homes and all. That is making of curd, wine. Then we learned about antibiotics and all. This already we learned, right? So microorganisms helps in the preparation of curd and all. So actually these all comes under what? Biotechnology. Biotechnology. So use of biological organisms and processes to manufacture products useful to humans. That is biotechnology, that is technology plus life or living things plus technology which is useful for human beings. And alcohol, the equipments, all the procedures, all the steps which all together it also comes in the biotechnology. Okay, so mainly what here you can see the European Federation of Biotechnology, that is the EFB, European Federation of Biotechnology, EFB. They defines biotechnology as the integration of natural science and organisms, cells, parts therefore, and molecular analogues for products and services. That means everything comes together. European Federation of Biotechnology defines biotechnology as the integration of natural science and organisms. Cells are there, parts thereof, and molecular analogues are there for products and services. Actually, biotechnology which mainly benefits and lots of research work as already mentioned is being going under this biotechnology, and day by day new things has been found under the biotechnology. And biotechnology deals with, already we know, that is microbe mediated processes which we are very familiar, making curd, bread, wine, etc. Then another term, that is in vitro fertilization, that is test tube baby program, or the cloning you have already heard, right? Then it also deals with synthesis and using of a gene. That means a selected gene being used or introduced like that. Then preparation of DNA vaccines. It also deals with correcting a defective gene. If at all any problem is there with any gene, and for correcting also we can use biotechnology. So it's a great scope for biotechnology in coming days, and great research works also going on. So these are the areas where it focuses. And the core techniques of modern biotechnology. Actually, these by basis of biotechnology includes classical biotechnology and modern biotechnology. Classical in the sense that it's making of wine, curd, bread and all, which we already know traditionally been used, right? But modern technology, modern, new upcoming modern technology is there, and in that the modern biotechnology, the process which include one is genetic engineering. Engineering in all other fields, civil engineering, mechanical engineering, we know, right? Here the engineering is being done in genetics. Genetics means gene. Gene means the DNA. So this here the technique is the technique in which genetic material, what are the genetic material? Yes, the DNA and the RNA. So that they are chemically altered and introduced to a host organism to change the phenotype. Here what is they are doing? They are selecting genetic material and they are altering them, and they are being introduced into the host organism to change the phenotype, to bring changes. Okay, so that technique altogether it can be called as genetic engineering. The technique in which genetic material is chemically altered and introduced into a host organisms to change the phenotype. That is genetic engineering. And the another one is bioprocess engineering. Bioprocess engineering means actually we develop antibiotics, vaccines, enzymes, etc., right? And it is being developed from the microbes and all eukaryotic cell. So here what the bioprocess engineering means, maintenance of sterile ambience, sterile, very clean ambience in chemical engineering processes for growing desired microbes. The desired high quality, high characteristics microbes will be grown in large quantity. Why we are growing that? To manufacture antibiotics, vaccines, enzymes, etc. So that process is known as, that is growing desired microbes and eukaryotic cells, and for the manufacture of antibiotics, vaccines, enzymes, etc. are known as bioprocess engineering. So these comes under the modern biotechnology, genetic engineering and bioprocess engineering. And here you can see the basic steps in genetically modifying an organism. So genetically, when we say genetically modified organism, so we need to simply, we cannot do. There are certain steps followed. So the important 3 steps followed for genetically modifying an organisms are identification of DNA with desirable gene. From that, by reading itself, you can understand identifying of DNA with desirable gene. That means some special characteristics should be selected. Then after selection of that desired DNA, what they have to do? They have to introduce that to a host. That is introduction of identified DNA into the host. And after introduction, after selection and after introducing, the job is over? No. We have to maintain it also. That means maintenance of introduced DNA in the host and transfer of that DNA to its progeny. Then only the full process is completed. That means the selected one should reach ultimately where? Should seen in the progeny, right? So these are the important 3 steps which for getting a genetically modifying or genetically modifying an organism. So we will discuss detail these 3 steps. So first one, identification of DNA with desirable genes. So traditional hybridization techniques leads to inclusion and multiplication of undesirable and desirable gene. Okay, that means olden method, what they do, they will select or they will, it's a technique which leads to inclusion and multiplication of both desirable and undesirable gene. That means both which is having good characters as well as both are being selected. But in genetic engineering, the modern technology, what they do, they select only the desirable genes for introducing. That means only desirable genes are introduced, not both undesirable and desirable. In genetic engineering, olden methods, both are being selected and both are being introduced. But in the case of genetic engineering, modern technology, only the needed characters or desirable one only introduced. So that is identification of DNA with desirable genes. And the next step is introduction of the disidentified DNA into host. We need to, that identified DNA should be introduced into the host. So here you can see an example given also. So the vector DNA, that means the selected DNA such as plasmid, is used to deliver an alien piece of DNA into the host organism. That means vector which carry, vector DNA means a plasmid which carry the selected DNA, which helps the selected DNA to be introduced into the host organism. So here you can see, so this is a bacterial DNA and this is a plasmids here. So what happens there? So here we need to introduce here in the host cell. This is the host cell. So this is being combined here, that is a recombinant plasmid along with the selected DNA. Okay, alien piece of DNA means the selected which we need to introduce to the host. So along with the vector DNA, we can see it here, and that is been introduced here in the E. coli cells, that is the host. And after introduction, you can see here what happened, the transformed cell which consists of the recombinant plasmid. So how they are introducing? They will select a vector DNA. Here an example is plasmid, and along with that the alien piece, that means the piece of DNA, the selected DNA will be mixed up and that is introduced into the host organisms. So that is the second step, introduction of the identified DNA into the host cell. And the third step is maintenance of introduced DNA in the host and transfer of that DNA to its progeny. So after introduction, it is not over. We need to see it in the progeny also. It should be transferred to the progeny. So a piece of alien DNA has a sequence called origin of replication, ORI, origin of replication. So need to start replicating, multiplying. So it cannot multiply itself in the progeny cells of organisms as they are an alien, they are being introduced, right? So hence this DNA is being integrated into the recipient genome. Recipient means which is the host genome, and recipient genome, and it multiplies and inherits along with the host DNA. So first what happened, they after introduction they cannot multiply or replicate by themselves. So along with the host, the recipient genome, it will be integrated and it multiplies and inherits along with the host DNA. So that then later on we can see it in its progeny also. And next comes the principles of biotechnology. So what is recombinant DNA technology? You might have heard at least what is recombinant DNA technology. So the process of joining and inserting a foreign piece of DNA into a host organism to produce new genetic combinations is called recombinant DNA technology. Just we have discussed the steps, right? So the process of joining and inserting a foreign piece of DNA, that means the selected desired DNA into a host organisms. For what? Why we are doing? To produce a new genetic combination, to get a more desirable characters. So that process can be called as recombinant DNA technology. So the first recombinant DNA technology, and its short form is small letter R, DNA, means recombinant DNA can be represented as small letter R then capital letter DNA, was produced by Stanley Cohen and Herbert Boyer in 1972. So first recombinant DNA was produced by Stanley Cohen and Herbert Boyer in 1972. So what did they do? So they isolated an antibiotic resistant gene. They selected or isolated an antibiotic resistant gene, that is a piece of DNA, from a plasmid of Salmonella typhimurium. It's a Salmonella typhimurium, and what did they do? It was linked with a plasmid of vector and transferred into E. coli. Antibiotic resistant gene, that is a piece of gene from Salmonella typhimurium, was selected and it was transferred into E. coli, Escherichia coli, and as a result what happened? The gene was expressed and multiplied in E. coli. That means here the process of inserting, which is the foreign DNA here selected, that is the antibiotic resistant DNA from where? That is Salmonella. And that what is the host here? That is E. coli. And what is the genetic combination? That means the gene was expressed and multiplied in E. coli. So this is the first work of recombinant DNA technology done by Stanley Cohen and Herbert Boyer. Okay, so to find out what antibiotic resistant genes. So this is all about the introduction and as well as the principles of biotechnology. I hope it is clear to you. So biotechnology, mainly life along with technology, living organisms and technology, advanced technology which is used in relation to living organisms, which mainly benefit or useful for the human beings. And we discuss about the main important steps in getting genetically modifying an organism, that is identification of DNA with desirable gene. After identification, second step, introduction of the identified DNA into the host, and third step, maintenance of introduced DNA in the host and transfer of that DNA into the progeny. Okay, so these are the main three steps to get the genetically modifying an organism. Then we discuss about the principles as well as the recombinant DNA technology. So this is all about the introduction and principles of biotechnology, and next we need to discuss about tools of recombinant DNA technology, which will be discussed in the next video. Thank you.